In the French countryside, after the Battle of Crécy in 1346, Benedictine monks carried the wounded from the field. They did not reach for vinegar. They did not reach for poultices of honey and ash. They went to the monastery garden and cut the thick, bristly leaves of a plant they had been growing for one purpose and one purpose alone. The soldiers healed faster, not anecdotally faster, not by the monk's subjective impression, measurably, documentably, repeatedly faster, over 2 centuries of battlefield medicine and across 3 monastic orders from France to England to central Germany. Those monks could not name the molecule responsible. They could not explain the mechanism. They just knew the plant worked, and so they kept planting it, kept harvesting it, kept pressing it against broken ribs and shattered arms. They called it knitbone. They called it bone set. They called it bruisewort. The scientific world took until 2023 to catch up. A peer-reviewed study published in a molecular biology journal that year identified the precise compound in Comfrey root that activates osteoclasts, the specialized cells your body recruits to remodel and reconstruct damaged bone. It named the compound, traced its pathway, and measured its effect on fracture defect models in living tissue. The compound is allantoin. The plant is Comfrey, and it grows in the ditch at the end of your street. What every pharmaceutical company, every calcium supplement manufacturer, and one specific federal agency does not want you to understand is this: calcium does not build bone. Calcium is the raw material. Your body must first send osteoblasts and osteoclasts to the fracture site, clear the cellular debris, lay down a collagen scaffold, and only then does mineralization, the incorporation of calcium, begin. Without that scaffolding process, calcium in your bloodstream is irrelevant to your broken bone. It just circulates. Allantoin accelerates the scaffolding. It binds to receptors that trigger osteoclast maturation. It upregulates Rankle signaling, the biochemical signal that tells your body to actively remodel the fracture site. It stimulates osteoblast proliferation, and it promotes the fibroblastic activity that lays down the collagen matrix that mineralization then follows. In other words, calcium heals bone the way bricks build a wall. Allantoin is the architect. It shows up first, reads the damage, and organizes the reconstruction crew. In 2008, a study found that administering Comfrey root extract produced measurable increases in radiographic bone density around titanium implants during active healing. The bone was not just knitting, it was knitting denser than controls. One compound in a plant that costs nothing, that you cannot stop from growing. The taproot of a mature Comfrey plant drives between 2 and 4 feet into the earth, deeper than any agricultural crop, deeper than most ornamental trees, deeper than the mineral-depleted topsoil layer that conventional farming has stripped bare over the last 80 years. Down there it finds what the surface cannot offer: potassium, phosphorus, nitrogen, calcium, magnesium, boron, manganese. It pulls all of it upward into its broad, hairy leaves. Cut those leaves to the ground and the plant regrows from that root within weeks, harvestable 4 times per season, every season, for decades, without fertilizer, without irrigation, without intervention of any kind. This is why Comfrey grows as a weed. It does not need you. Every root fragment left in disturbed soil becomes a new plant. Every seed that drops near a garden edge germinates with no assistance. It colonizes roadsides, riverbanks, abandoned lots, and field margins across North America, Europe, and Central Asia, because it evolved specifically to grow in disturbed ground, and human civilization produces nothing but disturbed ground. The plant does not know it is free. It is simply doing what it has always done. The people who first understood this were not monks. They were earlier. By 400 BC, Greek physicians had documented Comfrey's bone healing properties. Herodotus had already recorded its use for stanching severe bleeding. Nicander, writing in the 2nd century BC, listed it in his Pharmacopoeia of Antidotes. Galen, the physician whose medical writings dominated European practice for 1300 years, described its wound-healing action and prescribed the root externally for fractures. But it was Dioscorides, physician to the armies of the Emperor Nero, who traveled across the entire Roman Empire, carrying his botanical knowledge into military campaigns, who documented Comfrey most precisely. He wrote that the root, applied as a compress, sealed fresh wounds. He wrote that it had a joining together effect when cooked with flesh. He wrote this in the 1st century AD in 5 volumes that remained the foundational medical text of Western civilization until the 17th century. For 1600 years, the consensus did not change. Saxon herbalists recommended it. Cistercian monks grew it specifically for battlefield use. Hildegard von Bingen, the 12th century abbess whose botanical medicine texts are now studied in pharmacology programs, wrote that comfrey healed wounds no other plant could reach. Medieval European battlefield surgeons used it first when nothing else was available. They used it because it worked faster than anything else they had. None of them knew about allantoin. They knew what they saw. The first modern pharmacological confirmation arrived not in a splashy headline, but in the slow accumulation of clinical evidence across the 20th century. Allantoin was isolated and identified as the primary bioactive. Comfrey root extract was standardized. Randomized controlled trials began in the 1980s and accelerated through the 1990s and the 2000s. The results were not modest. In a single-blind, randomized, parallel group, multi-center clinical trial published in Phytomedicine, 164 patients with acute ankle sprains received either comfrey root extract ointment or diclofenac gel. Diclofenac is one of the most widely prescribed pharmaceutical anti-inflammatories in the world, the drug that sits in every sports medicine kit from Sao Paulo to Seoul. The trial was designed to demonstrate non-inferiority of the comfrey extract. The re-evaluation showed superiority. In the primary variable, measured pain response under calibrated pressure, comfrey root extract outperformed diclofenac gel, with a statistical significance of p equals 0.0012. Not comparable, not equivalent, superior, pain at rest, pain on movement, joint swelling, rate of recovery, comfrey outperformed or matched the pharmaceutical in every measured variable. The study authors recommended it should be part of physicians' standard treatment toolboxes. It is not in physicians' standard treatment toolboxes. In July 2001, the United States Food and Drug Administration, FDA, issued a letter to every dietary supplement manufacturer in the country. The letter did not discuss the clinical trial evidence. It did not mention the 40 published studies. It named one concern and one concern only: pyrrolizidine. Pyrrolizidine alkaloids, or PAS, are compounds found primarily in the root of comfrey that, when consumed in large quantities over extended periods, have been associated with hepatic veno-occlusive disease, a serious liver condition documented in livestock and in humans who consumed concentrated comfrey root tea daily for months or years. The FDA advised manufacturers to pull all comfrey products from shelves. Canada proposed a ban. Australia followed. Germany imposed restrictions. Within 18 months, the plant that 1,600 years of medical consensus had called knitbone had been reclassified in the regulatory vocabulary of the modern pharmaceutical state as a liver toxin. What the FDA letter did not say is worth measuring. Pyrrolizidine alkaloids are concentrated overwhelmingly in the root. The leaf contains far lower levels. The mechanism of harm, cumulative hepatotoxicity, requires prolonged oral ingestion of concentrated root material, not topical application, not leaf infusion, not the external poultice that represents 2,000 years of documented comfrey use. Not one randomized controlled trial on topical comfrey application has documented liver toxicity. Not one. The European Medicines Agency, EMA, assessing the same compound in the same time period, reached a different conclusion. They approved topical comfrey root preparations as a licensed herbal medicine for musculoskeletal pain and inflammation. Several EU member states certify comfrey root extract cream for clinical use to this day. The FDA banned internal supplements. The EMA licensed topical preparations. The plant did not change. The risk profile did not change. The regulatory outcome was not the same. What changed was jurisdiction. Meanwhile, the science continued. The 2023 PubMed study on allantoin and osteoclast activation did not cause a policy reversal. The comfrey root extract cream that outperformed diclofenac is not available on American pharmacy shelves. The plant that grows for free in the disturbed ground beside every highway in the continental United States remains, in the vocabulary of the American regulatory system, a liver toxin unfit for therapeutic use. The monks at Cressy did not have a regulatory agency. They had broken soldiers and a garden. The garden worked. Here is what the research, the tradition, and the clinical evidence together establish. Comfrey is not a calcium supplement. It is not trying to replace calcium. It is doing something calcium cannot do, activating the biological machinery that fracture repair requires before mineralization can begin. Two compounds do the primary work. Allantoin drives cell proliferation, osteoclast maturation, and collagen scaffold construction. Rosmarinic acid, present throughout the plant, suppresses the inflammatory cascade that slows healing and amplifies pain, reducing pore edema by over 60% at therapeutic doses in direct pharmacological testing. Neither compound requires a prescription, neither requires a laboratory, neither requires a supply chain. If you grow comfrey in your garden, and in most jurisdictions you can because topical use was never banned, a mature plant delivers cut material 4 to 5 times per year from the same root system, indefinitely. Fresh leaf applied as a poultice over a fracture site, a bruised joint, or a torn ligament delivers both allantoin and rosmarinic acid transdermally, exactly as battlefield surgeons from 3 different centuries documented. If you want to find it without growing it, drive the rural backroads of any temperate American state and scan the drainage ditches in spring. The broad leaves emerge in March before most other plants. The clusters of small purple-pink flowers follow in May. The root mass beneath them is already 3 years old, already mining the subsoil for minerals, already concentrating allantoin at levels no pharmaceutical synthesis can match for cost. It costs nothing because it needs nothing. That has always been the problem. A 2021 analysis in the Pharmaceutical Journal concluded that modern clinical data fully substantiates the traditional topical application of comfrey root preparations. 33 citations, decades of randomized trials. The conclusion was the same conclusion the Cistercians reached when they planted it in every monastery garden in the Holy Roman Empire. We did not lose this plant. We still walk past it every day. We just stopped recognizing what we were walking past. Every like and every share you give this video moves it past the algorithm to reach someone who needs it. The next vault opens soon.